looking through a glass. And in there is a character called Red Queen appearing in there. So this Red Queen theory is about that. I'm going to be explaining at the next couple of slides. Just to give a brief message to a young scientist to be in the future. Today's talk pretty much involves a lot of microscope images or videos, and I'd like to convey in my talk the message that the power of seeing observation is really enormous. If you really think about back in 1950s, when people really didn't know how the cells are dividing, there was a theory called there is a string-like shape called mitotic spindle, which seemed to segregate some of the important part of the cells. And there was a huge discussion. Nowadays, we know there is a mitotic spindle. It's described in the science textbook, and even for the elementary kids, everybody knows about it. But back then, in the 1950s, top researchers say, let's correct, 10 of the top researchers, 8 or 9 of them were denying it, rejecting the idea of the existence of mitotic spindle for various reasons. And then there came one guy, Shinya Inoue, who developed a new microscope, and in front of everybody showed, hey, there is indeed this mitotic spindle. And then all of a sudden, over one night, this contradictory discussion stopped. And of course, since then, it's established that there is such a structure in the cells. So that's the power of seeing. Even though the top researchers say the other things, you can just show one image, and then all of a sudden, the science changes. And the microscope is that powerful, but you have to be really carefully using it. Here is a picture from the picture book of the Alice in Wonderland. That's book 2, looking through the glass, Red Queen. So in this fantasy land, Alice is trying to make a progress by trying to move forward rigorously, but she goes nowhere. She never proceeds. And Red Queen comes in, starts to talk to Alice and says, hey, Alice, in this kingdom of mine, you have to run as fast as you can in order to keep one place. And this anecdote really became very popular in biologists to explain some of the very important phenomena in the evolution biology. I'm not going to be going too much into the details about it, but if you think about the relationship between the predator and the prey, they both keep evolving, one character after another. And if they stop the evolution, the prey will be overcome by the predator and then become extinct. And the story also applies to the opposite direction, vice versa. So that each of the species have to keep evolving at their full throttle in order to really maintain the status quo. And the second part is about the sexual reproduction versus asexual production. I'm not going to be talking about it, but there has been an unsolved mystery about that aspect of evolution biology. And this Red Queen theory beautifully explains such unsolved mystery. Now, today I am going to talk about Red Queen theory. Nobody really thought about it in our field in basic cell biology, but there is a very important mysterious phenomena in our field which nobody really previously appreciated, but it has really casted such a huge mystery. And this theory also seems to explain beautifully some of the mysterious behavior of the cells about the microtubules. I'm going to be explaining it later. Microtubules. I'm going to be spending the next few slides about explaining about microtubules. In the human society, you have highways, of course. You need highways, right? Without highways, what happens to the human society? People will really become crazy because think about the holiday season. They need the Amazon gift, right? The Amazon trucks cannot probably move efficiently with the highways, and people probably become crazy and start to blame each other, and the human society might collapse. So we need the highways. The same thing applies to the cells. In the cells, in order to really efficiently transport things, cargoes back and forth, they need the highways. And this red thing is the highways, and the green thing is the cargo, like Amazon trucks. And if you look at one particular part of the cells, the cargo is moving along the highway. So that is the red thing is the microtubule network. So microtubules really serves as a similar thing to the human society's highway. However, there is one huge difference between the human society highway and the microtubules. And nowadays we can beautifully label the cells in the live fashion, how the highways are moving here and there. The loom is too light, so probably some of you back there may not be able to decide it, but if you look under the microscope, it's really beautifully labeled. So the big difference between the human society highway and the cellular highway, which is microtubule, is that once we build the highways, we keep using it again and again and again, right? We don't seldomly break down the highways. Once we build it, that's it, right? It exists there almost forever, which is not the case in the cells. It keeps constantly being synthesized and being degraded. And the intactness or healthiness of the highways really depends on the balance between the synthesis and the degradation. And luckily enough, we can monitor the speed of microtubules being synthesized by labeling some of these construction vehicles in the cells. There are also construction vehicles, which is really making the cellular highways. And here we have labeled some such construction vehicles of the highways. Just during the time they're synthesizing the microtubules in the green color. Yeah, can you see these dots moving back and forth? So that's the power of seeing. We can literally monitor the speed of cellular microtubules being synthesized in such a way. And finally, using this system, we can compare between the healthy cells control and the disease cells. See here, we mimicked the burn injury. We collected the burn serum from the mouse and incubated the cells in the serum. And unfortunately, I just found out that this video was somehow so corrupted and it doesn't probably play here. But here in some other disease model, mimicking human disease condition called sepsis, we incubated cells in the presence of endotoxin. And top healthy cells, you can probably see the white dots moving quite rigorously. Can you? Once again, you may probably need strong pieces of glasses. On the top panel, the healthy cells are really synthesizing the microtubules, the highways, quite rigorously, while the bottom disease cells are doing such a poor performance. And I didn't show you in the previous picture, but the same thing was happening in the control arm. And in some other disease model called oxidation stress, which represents many human diseases, or some of the aging process also is linked to the excessive oxidative stress, you will be seeing exactly the same phenomenon. The healthy top cells are showing quite rigorous microtubule synthesis, while the bottom oxidation stress, oxidative stress, cells put under such stress is really showing such a poor highway construction. What happens after that is the big question to us. Highways are not being synthesized, being constructed so rigorously, contrary to expectation. And surprisingly, if you just look at the highways itself at rest, the highway construction isn't really that difference between the healthy cells on top and unhealthy cells on the bottom. Despite the fact that the highways are so poorly constructed, the highway construction itself is maintained relatively well even in a disease condition. Well, if you are not really carefully thinking, you might probably stop your experiment here and, hey, my hypothesis, rejected. I expected maybe poor synthesis, the construction of the highway might lead to the destructed highway or something. But actually, if you somehow bring the cells to a condition where the society might require more highway demand, was the next question. What happens if, let's say, highways are needed? Let's say around this time of the year, Christmas season, we need a lot of highway traffic. And all of a sudden, this still maintained highway really becomes so disrupted. The red color is the actual thing, the bottom color is computer generated, the highway mapping, and it's almost gone. So you may probably dig one layer deeper into the analysis, and finally, you can really reveal what's really hidden in the mystery of the cell's behavior there. And also, we did another disease model of healthy cells and the sepsis model, which is a severe infection. And the normal circumstances at rest, although the highway synthesis is also really disturbed in the disease model, the highway construction or networks seems to be relatively intact. And all of a sudden, when you really look at the stress condition, like where the highway demand is upregulated, that relationship becomes all of a sudden disturbed. Highway is gone. Same thing with oxidation stress and so on. So here, I'm just giving two messages. You have to probably carefully observe it, and also you have to use a very carefully designed disease model or the models to really answer some of the questions. So what these data really gives us as a message is that the red queen theory here in the microtubule world. In the Alice's anecdote, Red Queen was saying to Alice, in this country, in order to really keep one place, you have to keep running at your best, right? In order to keep your status quo, you have to really try your best. In the cell biology world, in order to keep the microtubule network intact at the status quo, especially at the increased demand for its usage, you have to keep synthesizing the microtubule at its full throttle. Otherwise, under heightened demand, it will be gone. The highways are no longer there. Highways are really disrupted. And since we have been talking whether highways, human society will become crazy. So in the next few slides, I'm going to be showing you how crazy the cells become without the highways. Okay. Here, from here on, I'm going to be speaking some other new terminology called autophagy and mitophagy. How many of you have ever heard of these two words, autophagy, mitophagy? How many of you have never heard of autophagy, mitophagy? Okay. At some point, some of you might have heard of these words at least, because I think it was 2016, was it? Some guy won the Nobel Prize for the discovery of the intricate mechanisms of the autophagy mitophagy. So what it is doing is that the autophagy or mitophagy is eating the cells, cellular component itself, and digesting it. And in order for its proper function, these eating machinery or engulfing machinery have to transport within the cells under an efficient condition. And for their transport, these autophagosomes or mitophagosomes, this eating cargo has to travel alongside the microtubule network. Microtubule network is the red thing that I've just shown you. So with intact highways, they can travel very efficiently and meet with each, other cargos and do the processing thereafter. Without such microtubules, they can transport the cargo to its, shipping direction, or cannot meet with other cargo and so on. So a lot of bad things will start to happen in terms of autophagy and mitophagy. And here is the video image. All of you see it. On the panel, you have a healthy cells where the red thing, microtubules network is labeled in red, and the cargo, Amazon trunks are being labeled in green. And in this case, the red is microtubule, and the green is autophagosomes and mitophagosomes. And as see, these autophagosomes and mitophagosomes are quite robustly and efficiently moving alongside these microtubule network, cellular highway. While in the bottom, the disease conditions, it's static. It seems so poorly moving. It's not that it's not moving, it's surely moving, but it just gets really jammed. Traffic jam is happening here. Of course, that really doesn't seem natural. So obviously here we are detecting a new phenomenon, cellular poor transport. But just imagine if you're not seeing this video image, but just capturing the image and seeing that static image, you will never ever reach this kind of understanding because the numbers of the cargo seems to be all the same and distribution are not that different and so on and so on. So you need a live powerful observation in order to really reach some conclusion. And same thing with other, burn model. The healthy cells, autophagy, mitophagy, vigorously moving, and the burn condition, it's relatively jammed. And forgive me, I'm going to be showing only one color, but same thing, the control healthy cells moving so rigorously. The disease condition of the severe sepsis or the severe infection shows poor motion. These phenomena perfectly matches with the red queen theory and microtubule network disruption, so on and so on. So in order for these autophagosome and mitophagosomes, they have to move rigorously, and that is not happening in the disease conditions. So what's the outcome? So in order for the autophagy and mitophagy to complete its cycle, they have to move along and they have to complete it. So it's probably the autophagy mitophagy flux cannot be completed. So next we did the autophagy mitophagy flux analysis. And here I'm not going to be explaining too much detail, but showing you how efficiently the mitophagy is being processed in one cell and the other cell. Here on your left side, you have healthy cells, which is showing normal, healthy, robust mitophagy flux turning over. Mitochondria is being really processed normally, while the other the severe sepsis model, it's so much disturbed because of the traffic jam. It's not being processed properly, and therefore everything remains there. In this experiment condition, the quick removal of this disease mitochondria is the normal response, which is really lacking here in the disease. So the outcome is really severe, and so on. So we really did all these three models, the same exact finding we obtained. Why autophagy, mitophagy, normal processing is so important, and what happens if it's really so poorly defective in the disease model? And in order to really just give you a final concluding remark, let me introduce our view of autophagy, mitophagy in the cellular world. In the human society, you have nuclear power plant, nuclear power plant. And it is very important because in order for the electricity to being produced, you need very often nuclear power plant. But just imagine what happens if one accident or one disaster hits that nuclear power plant. You probably remember 3 Mile Island, depending on your generation. You might have heard of Chernobyl accident in Russia, Belarus, and the Ukraine, and Fukushima nuclear power plant accident in Japan when the big tsunami and earthquake hit. Once such a disaster happens, what happens is nuclear power plant starts to spill all this toxic radioactive waste to the environment, and which is really a risk to human society. People around that nuclear power plant, polluted air will get cancer and all these sorts of health issues. So, unfortunately, human society do not know how to handle this spilled nuclear waste. But in that cells, we have a perfectly beautiful system to really tackle these disturbed mitochondria. which would be otherwise be spitting a lot of toxic substances into the cellular environment and yielding the cell death. And that part is being taken care of by this autogene mitogen mechanism. This is pretty much the last slide. We have discovered a very new, unique phenomenon, which seems to be explaining a lot of contradictory findings and unsolved mysteries in our field of muscle biology or the critical illness, severe burn injury, and so on. So maybe it may have to do something with the aging process. And this autophagy mitophagy defect, now we understand, seems to have a certain specific pinpoint that we could probably be targeting with some drug intervention. So that's really the beauty of seeing the cells in the really intricate way with a carefully designed observation system. I really thank all my colleagues and some of them are here who have helped us a great deal. So that's it. My message is that autophagy mitophagy is really important thing. You know, to really know some of the mysteries or unsolved questions. If you want to answer those things, you have to really have a powerful monitoring tool and carefully designed disease model. Even if the one layer of experiment gives you a negative result, try to think deeper and plan some backup experiment which might dig one deep or two layers deep further down, and that will probably give you some of the beautiful answers. And in fact, I shouldn't be addressing that, but we have already discovered some of the really